Welcome back to the OpenHBI course Code of Life. This time we want to focus on the lifecycle management, so-called replication. And this is important because we want to understand how cells move forward, how they grow, how they replicate, and how they reproduce themselves. And this is important because each of the cells has a certain lifespan, and after that they will divide into two cells. Therefore, let's have a brief understanding of the cell cycle. And the cell cycle consists of two major phases. The first phase, and you see it on the right-hand side, this is the yellow one. This is the so-called interphase, and the interphase has some subphases, so to say. The first phase is the G1 phase. This is where the cell grows and creates cell organelles. And sometimes it says, okay, we are ready. We go back to the gap 0 phase, or the so-called resting phase. In that moment, the cell stops the division and temporarily or forever. This is sometimes necessary, or at the end of the life cycle, it will get the information for apoptosis, and then it goes into the cycle. After the G1 phase, you have the synthesis phase. The synthesis phase is important because DNA here is replicated, and it is important to have the same information available in both daughter cells. And the G2 phase is important for producing all proteins which are involved in the following mitosis phase. And this is all part of the interphase. And the mitosis phase, you see it on the top, indicated by the M. This is actually the phase when the cell really divides into two daughter cells. And we want to have a closer look how this cell division looks like and how does it work. Therefore, I brought some drawings here from a very popular book. It's called Henry Gray's Anatomy of the Human Body. It was published in 1918, and you most probably know Gray's Anatomy is the short version of that book's title. So, and everything which was drawn in these times, so these figures are actually from the book, is still accurate for the today understanding of how mitosis works. So first of all, we have the phase called prophase. In the prophase, you see something here called chromatins, and they condense into chromosomes. You see these kind of wired structure gets more and more structured to chromosomes. So these are typically these X's, you know, when you look for them. And the second very important thing is here the nucleolus disappears. So the structure in the middle of the cell core disappears because now the DNA flows around. And the third important component which is created here is the so-called spindle apparatus. There are two spindle apparatus because they are important for both cells later on. They move to the so-called poles of the cell. So one is going to the north and one is going to the south. Here you see it right and left. So they prepare already the division into two cells. So in the end of the prophase, you have chromosomes without any cell core any longer. The next phase, it's called metaphase, and within the metaphase, these chromosomes line up along that equatorial plane. And I added this red dotted line. This is so say the equatorial plane, where all these chromosomes are somehow originated there. And it's important because they are connected now also to the spindulus apparatus. The next thing is it's the so-called anaphase. And in the anaphase, these chromosomes are broke up in the middle of there at the equatorial end and moved into individual chromatids. So we have one chromosome which is now split up into two separate parts of the chromosome. And these individual parts of the chromosome are moving to the poles here, to the left and to the right. And you see they are moving and moving by the spindulus apparatus, which is important for splitting them up. And the last phase in the mitosis is the so-called telophase. The telophase is important to build again cell membranes to reappear the nucleus, and then you have again these individual structures which are unwind into DNA, which is more protected. So the chromosomes disappear and get into the chromatin state. So in the end, what you have as a result, you see you have two kinds of membranes. You have two cell cores, and in both of these cell cores you have parts of the DNA which need to be replicated now because you have only 50% of the chromosome in each of the daughter cells. But in the end, at this stage you have two individual cells again, right? Keep in mind you have two cells at that moment, but now you need to duplicate the information in the cell cars. And how does it look like? How does this replication look like? So the replication is done within the synthesis phase of the interface. And you see it's pretty complex, and therefore this is a very popular picture here. I don't want to focus too much on every of the details, but I'd like to introduce how does it work. So in the end you have one kind of DNA on the right-hand side, and this is split up into two parts with the same information. And how does this work is part of the replication. First of all, there are again three kind of phases. It's the so-called initiation phase. In this initiation phase, you have the individual enzymes which are involved. The first, and this was the big green one, this is the so-called topoimerase. And the topoimerase is important because it unwinds the DNA from this compressed version to a kind of long strand, and it is also able to overwind it again. So this is important because in the overwind structure, DNA is really protected. In the unwind version, it is really a problem because external mutagenes or any kind of triggers could change kinds of DNA. This is a risk for variants or mutations at that stage. The next important enzyme is the so-called helicase. This is important. This is the blue arrow here. This unzips the DNA at a specific origin and puts them into two separate strands, right? So this is typically also done in laboratories if you want to split DNA. And the third important enzyme here is the primase. It is added to the origins to bind polymerase. This is the blue circular format here. This is important because this gives you the start points for the polymerase, and the polymerase is important in the next phase. And this is the elongation phase. In the elongation phase, you will see that the splitted DNA grows again, and therefore the DNA polymerase is important. It first extends the DNA using a kind of template strand, and this is exactly the remaining strand, and the polymerase copies the compartment which is binding to the other side and produces it there and puts it at that point. The polymerase works typically in only one direction, 5' to 3'. And this is important because on the other stage you have one so-called leading strand, and the leading strand is in the right direction. That means the polymerase starts at a point and moves until the end. On the other side, you have to have multiple points where polymerase starts because you have only short distances where it works and then it needs to restart and restart. This is the so-called lagging strand. The third stage in this process is the termination. And at the termination, you have exactly done all the individual steps. You were able to reproduce the DNA and bring the pieces together. This is typically triggered by certain enzymes as well. So what to take home from this important lecture? First of all, mitosis results in two daughter cells carrying the identical DNA. This is most often the time. And the very important thing here is to understand how does it work, because how to ensure that these are identical. Therefore, the DNA polymerase performs a so-called proofreading. So every time it replicates strands, it can check if the replicated strand is really the inverse of the template strand. This is also the reason why the throughput of the DNA polymerase is different across eukaryotes and prokaryotes. In humans and eukaryotes, you see it's about 50 to 100 nucleotides per second, which is not fast. In prokaryotes, we are talking about thousands or even more per second. The reason is really that proofreading, which is time-consuming but very important to keep the DNA information stable. And therefore, how to reduce the overall time? The DNA is replicated in parallel. It's very similar to what computer scientists are doing. They use parallel computing to improve the throughput of the programs. Here, the DNA replication is parallelized. And it starts at different locations, and therefore you have only a pretty short amount of DNA which is unwind for a very short time. It's replicated, and just after that it's overwinded again. So therefore you can keep it in that protected, overwinded region and stable for most of the time. So what's next in the course setup? So next time we want to talk about how to use this encoded information in the DNA to produce something out of it. So to say, how to use the source code to build some programs out of it. And therefore, we want to investigate how these, you see it on the right-hand side, these ribosomes look like and how they work. And therefore, it's very important to have this crucial understanding of how the DNA looks like before we can turn over to this. Therefore, stay tuned, and I'm looking forward to see you next time.